Okay, well, let me just make sure. Hello, hello. Here we go. Fantastic. So, hey, it's Dmitry again, and I just streamed a chapter not that long ago, earlier today, but I figured I'd stream this one as well. I have certain things coming up, as you can imagine, and so if I have to read it, well, then I should probably stream it as well. And so that's what I'm doing here. We're working with, again, Guyton Hall, Pocket Physiology. Now we're on chapter 35. Resistance of the body to infection, 2, immunity and allergy. And again, that's basically from the Pocket Physiology textbook. If you wanted to follow along in the Pocket Physiology textbook, you can do that, or you can follow along with the actual Guyton Hall standard textbook. Again, that's resistance of the body to infection, 2, immunity and allergy. Let's begin. Innate and acquired immunity. Immunity is the ability to resist organisms or toxins that damage the tissues of the body. Most organisms have innate immunity, which consists of general actions such as phagocytosis of bacteria, destruction of pathogens by acidic secretions, digestive enzymes in the GI tract, resistance of the skin to invasion, and certain chemicals in the blood that attach to foreign organisms or toxins that destroy them. Acquired immunity is the ability to develop powerful protective mechanisms against specific invading agents such as lethal bacteria, viruses, toxins, and even foreign tissues from other organisms. Acquired immunity is initiated by antigens. Two basic types of acquired immunity occur in the body: humoral immunity, or B-cell immunity, involved in the development of circulating antibodies that are capable of attacking an invading agent. Cell-mediated immunity, or T-cell immunity, is achieved through the formation of large numbers of activated lymphocytes that are specifically designed to destroy the foreign agent. Because acquired immunity does not occur until after an invasion by a foreign organism or toxin, the body must have some mechanism for recognizing the invasion. Each invading organism or toxin usually contains one or more specific chemical compounds that are different from all other compounds. These compounds are called antigens, and they initiate the development of acquired immunity. For a substance such as a polypeptide to be antigenic, it usually must have a molecular weight of at least 8,000 kilodaltons. However, immune responses can also be generated against smaller substances called haptens. If they are attached to a larger carrier, such as a protein, the process of antigenicity depends on the regular occurrence on the surface of the large molecules of molecular groups called epitopes. Proteins and large polysaccharides are almost always antigenic because they contain this type of stereochemical characteristic. Lymphocytes are responsible for acquired immunity. Lymphocytes are found in lymph nodes and in special lymphoid tissues such as the spleen, submucosal areas of the GI tract, and bone marrow. Lymphoid tissue is distributed advantageously in the body to intercept invading organisms and toxins before the invaders can become widespread. There are two populations of lymphocytes, T lymphocytes and B lymphocytes, both of which are derived from pluripotent hematopoietic stem cells that differentiate to form lymphocytes. T lymphocytes are processed in the thymus gland and are responsible for cell-mediated immunity. So just remember T for thymus. B lymphocytes, which are processed in the liver during mid-fetal life and in the bone marrow during late fetal life and after birth, are responsible for humoral immunity. And so for B cells, just remember that the B lymphocytes are made in the bone. Okay? The thymus gland preprocesses T lymphocytes. Lymphocytes divide rapidly and develop extreme diversity for reacting against various specific antigens in the thymus gland. The processed T cells leave the thymus and spread to the lymphoid tissues throughout the body. Most pre-processing of the T lymphocytes in the thymus occurs shortly before and after birth. Removal of the thymus gland after this time diminishes but does not eliminate the T lymphocyte system. Removal of the thymus several months before birth, however, prevents the development of a all-cell mediated immunity. The liver and bone marrow pre-processes B lymphocytes. Much less is known about the details or processing of B lymphocytes. In humans, B lymphocytes are pre-processed in the liver during mid-fetal life and in the bone marrow during the late fetal life and after birth. B lymphocytes differ from T lymphocytes in that they actively secrete antibodies, which are large protein molecules capable of combining with and destroying substances. B lymphocytes also have a greater diversity than do T lymphocytes, forming millions and perhaps even billions of antibodies with different specific reactivities. After processing, B lymphocytes migrate to lymphoid tissues throughout the body where they lodge in locations near the T lymphocyte areas. When a specific antigen comes in contact with the T and B lymphocytes in the lymphoid tissue, a set of T and B lymphocytes become activated to form activated T and activated B cells, which subsequently form antibodies. The activated T cells and newly formed antibodies react specifically with the antigen that initiated their development and inactivate or destroy the antigen. Preformed T and B lymphocytes await activation by an antigen. Millions of types of preformed T and B lymphocytes are capable of responding to the appropriate antigen. Each of these preformed lymphocytes is capable of forming only one type of antibody or one type of T cell with a single type of specificity. Once the specific lymphocyte is activated by its antigen, it reproduces wildly, forming tremendous numbers of duplicate lymphocytes. If the lymphocyte is a B lymphocyte, the progeny eventually secrete antibodies that circulate throughout the body. If the lymphocyte is a T lymphocyte, its progeny develop into sensitized T cells that are released into the blood, where they circulate through the tissue fluids throughout the body and back into the lymph. Each set of lymphocytes capable of forming one specific antibody or activated T cell is called a clone of lymphocytes. The lymphocytes in each clone are identical, and all are derived from one progenitor lymphocyte of a specific type. Specific attributes of the B lymphocyte system: humoral immunity and antibodies. Upon entry of a foreign antigen, the macrophages in lymphoid tissue phagocytize the antigen and present it to adjacent B lymphocytes. The previously dormant B lymphocytes specifically for the antigen immediately enlarge and eventually become antibody-secreting plasma cells. The plasma cells produce gamma globulin antibodies, which are secreted into the lymph and carried to the circulating blood. Formation of memory cells enhances the immune response to subsequent antigen exposure. Some of the B lymphocytes formed during activation of the specific clone do not form plasma cells, but instead form new B lymphocytes similar to those of the original clone. This causes the population of the specifically activated clone to become greatly enhanced. These B lymphocytes circulate throughout the body and inhibit all the lymphoid tissues but remain immunologically dormant until activated again by a new quantity of the same antigen. The cells of the expanded clone lymphocytes are called memory cells. Subsequent exposure to the same antigen causes a more rapid and potent antibody response because of the increased number of lymphocytes in the specific clone. The increased potency and duration of the secondary response are the reasons why vaccination is often accomplished through the injection of antigen in the multiple doses, with periods of several weeks or months between injections. Antibodies are gamma globulin proteins called immunoglobulins. All immunoglobulins are composed of combinations of light and heavy polypeptide chains. Each light and heavy chain has a variable portion and a constant portion. The variable portion is different for each specific antibody. It is this portion that attaches to a particular type of antigen. The constant portion determines other properties of the antibody, such as diffusibility, adherence to the structures in tissues, and attachment to the complement complex. There are 5 general classes of antibodies, each with a specific function: immunoglobulin Ig, M, IgA, IgG, and IgD, and IgE. So basically, M, A, G, D, E. IgG class is the largest and constitutes about 75% of the antibodies of a normal person. Antibodies act by directly attacking the invader or activating the complement system, which subsequently destroys the invading organism. Antibodies can inactivate an invading agent directly in one of the following ways: agglutination, in which the multiple large particles with antigens on their surfaces, such as bacteria or red blood cells, are bound together in a clump; precipitation, in which the molecular complex of the soluble antigens in antibodies becomes so large that it is rendered insoluble; neutralization, in which the antibodies cover the toxic sites of the antigenic agent; lysis, in which antibodies are occasionally capable of causing rupture of an invading cell by directly attacking the cell membranes. Again, I'm running a secondary program just to maintain a transcript, and so I'll be waiting for that to follow through. Here we go. And I'll repeat again. Lysis, in which the antibodies are occasionally capable of causing rupture of an invading cell by directly attacking the cell membranes. Although antibodies have some direct effects in destruction of invaders, most of the protection afforded by antibodies derives from the amplifying effects of the complement system. The complement system is activated by the antigen-antibody reaction. Complement is a collective term used to describe a system of about 20 proteins normally present in the plasma that can be activated by the antigen-antibody reaction. When an antibody binds with an antigen, a specific reactive site on the constant portion of the antibody becomes uncovered or activated. This activated antibody site binds directly with the C1 molecule of the complement system, setting in motion a cascade of sequential reactions. When complement is activated, multiple end products are formed. Several of these end products help destroy the invading organism or neutralize a toxin. Complement can stimulate phagocytosis by both neutrophils and macrophages, cause rupture of the cell membranes of bacteria or other invading organisms, promote agglutination, attack the structure of viruses, promote chemotaxis of neutrophils and macrophages, and induce the release of histamine by mast cells and basophils, promoting vasodilation and leakage of plasma, which in turn promote the inflammatory process. Activation of complement by an antigen-antibody reaction is called the classical pathway. Okay. Special attributes of the T lymphocyte system, activated by T cells and cell-mediated immunity. When macrophages present a specific antigen, T lymphocytes of the specific lymphoid clone proliferate, causing large numbers of activated T cells to be released in the same way antibodies are released by the activated B cells. These activated T cells pass into the circulation and are distributed throughout the body, where they circulate for months or even years. T lymphocyte memory cells are formed in the same way that B memory cells are formed in the antibody system. Upon subsequent exposure to the same antigen, the release of activated T cells occurs far more rapidly and much more powerfully than during the first response. Antigens bind with receptor molecules on the surface of the T cells in the same way they bind with antibodies. These receptor molecules are composed of a variable unit similar to the variable portion of the humoral antibody, but the stem sect of the receptor molecule is firmly bound to the cell membrane. Let me check something briefly. Yeah, that's fine. Antigen-presenting cells, major histocompatibility complex proteins, and antigen receptors on the T lymphocytes. T cell responses are extremely antigen specific, like the antibody responses of the B cells, and are as important as antibodies for defending against infection. Whereas B lymphocytes recognize intact antigens, T lymphocytes respond to antigens only when they are bound to specific molecules called major histocompatibility complex, or MHC proteins, on the surface of an antigen-presenting cell. So that'd be something like a dendritic cell or a macrophage, maybe, something like that. The three major types of antigen-presenting cells are macrophages, B lymphocytes, and dendritic cells. Well, what do you know? Dendritic cells are located throughout the body and are most effective in presenting antigens to T cells. The MHC proteins bind peptide fragments of the antigen proteins degraded inside the antigen-presenting cell and then transport them to the cell surface. There are two types of MHC proteins, MHC class 1 and MHC class 2. MHC class 1 proteins present antigens to cytotoxic T cells, and MHC class 2 proteins present antigens to T helper cells. So remember, MHC class 1, cytotoxic T cells or killer T cells, and MHC class 2, T helper cells. Antigens, which is, by the way, for reference, I want to go a little bit further than that. The killer T cells for MHC class 1 are CD8 positive, while the MHC class 2 type related to the helper T cells are CD4 positive. So in both cases, the MHC class multiplied by the CD4 or CD8 of the T cell will always equal 8. So if it's MHC class 2, well, then you know it's going to be CD4 positive, i.e. it's going to be a helper T cell. If it's MHC class 1, then you know it's going to be a killer T cell because it's CD8 positive. For reference, antigens on the surface of the antigen-presenting cell bind with receptor molecules on the surface of the T cell in the same way that they bind with plasma antibodies. So here's a little diagram depicting the interaction between the antigen-presenting cell and the T cell. As you can see, there's a little cell-cell adhesion protein, sure. The T cell receptor right there, the foreign protein, and the MHC proteins presenting that foreign protein to the T cell receptor. There is definitely, if you read the immunology textbooks and stuff like that, it's going to be a lot more specific than what this is presenting here. So just keep that in mind. But I'll read the figure for you. Activation of T cell requires the interaction of T cell receptors with an antigen, foreign protein, that is transported to the surface of the antigen-presenting cell by a major histocompatibility complex, MHC protein. Cell-to-cell adhesion proteins enable the T cell to bind to the antigen-presenting cell long enough to become activated. Several types of T cells and their different functions. The three major groups of T cells are T helper cells, cytotoxic T cells, and suppressor T cells. The function of each of these cells is quite distinct. T helper cells are the most numerous type of T cell in the body. T helper cells serve as regulators of virtually all immune functions. This task is accomplished through the formation of a series of protein mediators called lymphokines, which act on other cells of the immune system and bone marrow. T helper cells secrete interleukins, I should say, if you're speaking English, interleukins, IL-2 through 6, granulocyte monocyte colony stimulating factor, and interferon gamma. In the absence of the lymphokines produced by T helper cells, the remainder of the immune system is almost paralyzed. It is the T helper cells that are inactivated or destroyed by human immunodeficiency virus, HIV, acquired immunodeficiency syndrome, AIDS, which leaves the body almost totally unprotected against infectious disease. T helper cells perform the following functions: stimulate growth and proliferation of cytotoxic suppressor T cells through the actions of IL-2, IL-4, IL-5. Okay. Stimulate B cell growth and differentiation to form plasma cells and antibodies mainly through the actions of IL-4, IL-5, and IL-6. Activate the macrophage system. Stimulate T helper cells themselves. IL-2 has a distinct positive feedback effect of stimulating the activation of the T helper cell, which acts as an amplifier to enhance the cellular immune response. Cytotoxic T cells are capable of killing microorganisms through a direct attack. Because cytotoxic T cells are capable of directly killing microorganisms, they are also called killer cells or killer T cells. That's distinct, by the way, from NK cells, which are natural killer cells. So NK cells are a different thing. We're talking about cytotoxic or killer T cells. So surface receptors on the cytotoxic T cells cause them to bind tightly to the organisms or cells that contain their binding specific antigen. After binding, the cytotoxic T cells secrete hole-forming proteins called perforins. that literally punch large holes in the membrane of the attacked cells. These holes disrupt the osmotic equilibrium of the cells, leading to cell death. Cytotoxic T cells are especially important for destroying cells infected by viruses, cancer cells, or transplanted organs. Suppressor T cells suppress the functions of both cytotoxic and T helper cells. It is believed that the suppressor functions of the suppressor T cells serve the purpose of regulating the activities of the other cells so excessive immune reactions that might severely damage the body do not occur. Something to consider, it's basically one of these sort of negative or like modulatory systems to prevent the body from kind of having an excessive response to something. Okay. Tolerance of the acquired immunity system to one's own tissues. Role of pre-processing in the thymus and bone marrow. The immune system normally recognizes a person's own tissue as being completely distinct from that of invading organisms. Most of this tolerance is believed to develop during the processing of T lymphocytes in the thymus and B lymphocytes in the bone marrow. Although the mechanism of tolerance induction is not completely understood, continuous exposure to the self-antigen in the fetus is thought to cause the self-reacting T and B lymphocytes to be destroyed. They basically believe there's a sort of a selection method where cells that are, or I should say lymphocytes specifically, that are reacting to self-antigens in an inappropriate way are destroyed because you obviously don't want to have autoimmune reactions all the time. Failure of the tolerance mechanisms leads to autoimmune diseases, as I said, in which the immune system attacks the tissues of the body, such as rheumatic fever, in which the body becomes immunized against the tissues of the joints and valves of the heart. Glomerulonephritis, in which the body becomes immunized against the basement membrane of the glomeruli. Myasthenia gravis, in which the body becomes immunized against the acetylcholine receptor proteins of the neuromuscular junction. And lupus erythematosus, in which the body becomes immunized against many tissues. Lupus is not great. Allergy and hypersensitivity. An important but undesirable side effect of immunity is the development of allergies or other types of immune hypersensitivity. Allergies can be caused by activated T cells and can cause skin eruptions, edema, or asthmatic attacks in response to certain chemicals or drugs. For example, in some individuals, a resin in the poison ivy plant induces formation of activated helper and cytotoxic T cells that diffuse into the skin and elicit a cell-mediated characteristic type of immune reaction to this plant. Some allergies are caused by IgE antibodies. These antibodies are called reagents or sensitizing antibodies to distinguish them from the more common IgG antibodies. A special characteristic of IgE antibodies is their ability to bind strongly with the mast cells and basophils, causing release of multiple substances that induce vasodilation, increased capillary permeability, and attraction of neutrophils and eosinophils. Hives, hay fever, and asthma can result from this mechanism. And that concludes Chapter 35 of Guyton and Hall's Pocket Physiology textbook. Again, if you wanted to follow along, you can look at the Pocket Physiology textbook or follow along with your standard version of Guyton and Hall's Physiology textbook. That concludes the end of the chapter. Thank you very much for listening.